Hi everyone, welcome back to Advanced Higher Biology. Today we're continuing with key area 5, protein control of cell division, and we're moving on to part C, control of the cell cycle. So we have looked at stages of the cell cycle beforehand. What we're going to be looking at next is how that cell cycle, as the title suggests, is actually controlled. So you might remember, going back to National 5, we did discuss that if mitosis was just unchecked and unregulated, if there's just all this division going on and on and on, you're going to end up with uncontrolled cell growth, and that's where we end up with tumours. So what we're looking at here is how is the cell cycle actually controlled? How does the cell stop, look at what's happening, and then allow the rest of the cell cycle to progress? So first off, there are several checkpoints within the cell cycle, and what these checkpoints do is they assess the condition of the cell. They basically check that everything is working as it should and there's no abnormalities during the cell cycle. If there is an issue, then they're going to stop the process, but at each checkpoint, the progression is halted until certain requirements are met. So until the cell has assessed that there's nothing wrong going on. Now, while we go through this, you will come across some new terms. We'll talk about these G1, G2, S phases, and your M phase as well, which we will be discussing. So, first of all, when we look at how these checkpoints actually work, we have new proteins we're going to talk about called cyclin proteins. Now, during cell growth and within the cell cycle, there are a bunch of cyclin proteins, and their key role is to actually regulate this cell cycle. Now, cyclins are going to be combining with something called a cyclin-dependent kinase. So they're going to activate what we call CDKs, and these CDKs are going to be really crucial to allowing the cell cycle to progress. So what happens is an active cyclin CDK is going to phosphorylate proteins that then allow progression to take place. And if there's enough of that successful phosphorylation, then progression is going to occur. So if you're wanting the cell cycle to continue, you need to have your CDKs, phosphorylating proteins, hitting a threshold, and then we continue on. Now, what we're going to have, like I said, is various checkpoints. So at metaphase, if you remember where metaphase is in the cell cycle, this is going to control the progression from metaphase further on to anaphase. So remember, we start with a prophase, we then have metaphase, and then before we get to anaphase, we have that metaphase checkpoint. The progression is then halted, as it always is, until those chromosomes are lined up correctly on the metaphase plate. So remember, lined up at the equator, you have all the chromosomes in a nice line, and then they're going to be attached to spindle microtubules before they get pulled across in the process of anaphase. Now again, that's just one example of a checkpoint of what's going on. If, for example, the cell moved on to anaphase too quickly, you might only have some of the chromosomes being pulled apart, and you have a loss of genetic material, that side of things. Now, the next one that we're going to talk about a bit more, and is a bit more complicated, is what's called your G1 checkpoint. Now, there's going to be another word that we're going to come across here called retinoblastoma, commonly referred to as RB. And the role of RB is to act as a tumour suppressor, so to try and stop tumours from forming. Now, how it does this is it inhibits the transcription of genes that code for the proteins needed for DNA replication. So, simply put, your RB is there to inhibit the transcription of these genes that are going to create all these proteins that allow further replication of DNA. So, if, for example, your RB was not working, or if there was a mutation, which sometimes comes up in questions as well, of retinoblastoma, if it's not able to inhibit the transcription of those genes, then obviously transcription will take place, and it means there's going to be lots of DNA replication taking place. So again, this could be in a question to do with tumours and cancers. Now, what's going to happen in this process in the G1 checkpoint is phosphorylation by those CDKs that we talked about, the G1 CDKs in this case for this checkpoint, is going to inhibit the retinoblastoma protein. So phosphorylation by the CDK inhibits the RB, and then that then stops the inhibition of the transcription of genes. Okay, so this then allows the transcription of genes to now take place, so stopping that RB from working, like we mentioned. Then what happens is we're then going to have those proteins needed for DNA replication to actually produce these genes and move from G1 to the S phase where replication takes place. So this all seems like a lot of different abbreviations all thrown together. Just trying to remember, RB is there to try and stop the genes that we require for DNA replication to work, to be produced. If you have phosphorylation of CDK at the G1 checkpoint, though, that inhibits the RB from inhibiting. So it allows the transcription of those genes and DNA replication can take place. And try and ignore the huge amount of spelling errors in this slide. Next, you're going to have a G2 checkpoint. So after you've had DNA replication, after you've had that S phase, we now arrive at G2. Now, through the G2 checkpoint, again, you're going to halt the process, you're going to check what's going on, and this time we're going to look at the success of DNA replication. So, has DNA replication taken place? Has it taken place successfully? Is there any damage to the DNA? Now, if there is damage to the DNA, there's going to be an activation of several proteins, including one called P53. Now, P53 is able to basically fix the DNA. It stimulates DNA repair. But what it can do as well is it can stop the cycle, or it can lead to just full cell death. Okay, so again, the checkpoint is looking over the DNA replication, having a look what's going on. It's almost quality control before you move on to the M phase. If there's any damage whatsoever to the DNA, P53 comes in to either repair, arrest, or cause cell death and stop the cell cycle. Again, no, if everything's fine, this checkpoint is then passed, and then cell then progresses from the G2 to the M phase. Now, as we talked about before, it is an issue if your cell cycle is not being controlled, if it's unregulated, and you do need to know examples of these. So this is something we've touched on before, maybe a couple extra bits as well. Now, if there is an uncontrolled reduction in the rate of the cell cycle, so if the cell cycle essentially isn't completing fast enough, if it keeps getting stopped, if there's too much cell death going on and we're not reaching the amount of replication that we require, then that can result in degenerative diseases, so things like Parkinson's or Alzheimer's. So try and remember that a reduction in cell cycle leads to degenerative diseases, but the same is also true as well. If there's an uncontrolled increase, then we're going to have tumor formation, like I've mentioned before. So if there's too much cell division going on, you're going to result in uncontrolled cell growth, which is a tumor. And then what we have here is we're going to discuss a proto-oncogene. So proto-oncogene is just a gene that's involved in cell growth, but it can mutate to become a tumor-promoting oncogene. So like the word suggests, that is going to just start producing tumors, okay? And oncogene, you might recognize from oncology, studies of tumors and cancers as well. If you have too much cell growth, you're going to result in tumors. And that's all for 5C. So again, that's going through these checkpoints and the importance of them all. It's important for you to know things like your CDKs, your RBs, your P53s, and what their role is, because that's a common question that can come up. But also have a look at what would happen if RB was mutated. What would happen if there was not enough phosphorylation of CDK? And try and also remember your order of your G1, your S, your G2, and your M phase as well. Sometimes that comes up as a little multiple choice question. So that is all for part 5C, and we're going to finish off in the next video with 5D, control of programmed cell death, or stuff you may be aware of as apoptosis, to finish off unit 1. So hope that's been helpful for you. Take a look over that and make sure you know your different forms of proteins and your order of cell division, and I'll speak to you soon for 5D. Bye for now.